Good morning. Welcome to the Fireside Chat. My name is Brian Kane. I'm the co-founder and publisher of BioProcess International. And along with Ann Montgomery, who's the editor-in-chief and co-founder of BioProcess as well, we are your co-moderators for today's session. The theme of this discussion is bringing it all together for commercialization, perspectives from every stage. For many years, the biopharmaceutical industry has approached product development, upstream, downstream manufacturing, and the supporting analytical methods as a series of separate and independent steps and processes. Many companies' infrastructures have been designed to mirror this approach, with each department responsible for only achieving that specific and individual goal. It's certainly an approach that still works, but like many things that do work, it may not remain to be the best way or the most efficient way to achieve a goal. So in today's competitive competitive environment, the dynamics of biopharmaceutical development are constantly changing and evolving. Technologies are changing, the science is changing, the management expectations are changing. So working in process silos doesn't make as much sense as it used to. And that's okay, because like in every industry, biopharmaceuticals must continually evolve and adapt in order to move forward. In Tuesday's keynote, Rohini from Amgen mentioned that the average cost to bring a drug to market today is about $2.6 billion. And the average time to bring a drug to market is now about 128 months. That's really too much money and too much time to sustain success. So in order to mature and grow as a business, the biopharmaceutical industry must find a way to reduce overall development cost and time to market. The key is not to look at upstream and downstream and analytical as independent processes, but to mirror the format that Barry Walsh created for this conference and look at each phase of development as a complete process. And like marriage and parenting and life, communication is the key to making all things work. All departments must be on the same page and agree to what the ultimate goal is and how to achieve it. This will ensure the latest science and technologies are being utilized and synchronized in an approach which should improve efficiency and efficacy and ultimately lower drug development costs and time to market. So that's really the goal of today's conversation. Barry's put together a terrific panel here that conservatively has over 100 years of combined experience. These folks represent every phase of biopharmaceutical development and have seen what has worked and what has not worked. We have the opportunity this morning to tap into this and take advantage of this group and rely upon their unique experiences and expertise to find out and figure out what they've done, how they've done it, how they've overcome challenges, and apply it to your processes and your businesses. And if we can do that, we can help ourselves, we can help our companies, and ultimately we can help the patients upon which are relying upon us to create the life-saving drugs and treatments. So this is an interactive discussion panel. We're going to focus on four main categories: technology application, platform development, reducing development time, cost, and reduction. We'll ask each panel member the same questions, and then we'll provide you, the audience, the opportunity to join in on the conversation. So before we get started, let me just do a brief introduction of our panelists here. Paul Wu is currently Director of Upstream Development, Bayer Global Biological Development Organization. Paul has over 20 years of experience and is responsible for cell line generation and cell culture process development. Ali Siapush, at the end of the table, is currently president and founder of PharmEffects, a biopharma consulting company. He has over 20 years of experience working with Dendreon and Amgen, where he was responsible for early and late-stage product development. He has helped Dendreon's automated manufacturing platform and provided technical support for the commercial manufacturing. Gunter Yashies, currently Senior Director of Strategic Customer Relations at GE Healthcare Life Sciences, has published countless articles and has published a number of books, including Process Chromatography Handbook in 2008 and the development of a therapeutic monoclonal antibody products in 2010. In addition, in 2012, Gunter was awarded the BioProcess International Award for Thought Leader of the Decade in Downstream Processing. And finally, Catherine Chalupka is currently a senior scientist at Amgen, and she is responsible for commercial development, process development, and characterization from phase 2 through commercial. So with that being said, I'm going to turn the microphone over to Ann Montgomery, and we're going to get the conversation and the questions started. Ann.
I can maybe add to that from a kind of technology development perspective on the vendor side. There is one example of a technology, a major one that we developed in the early 90s. The commercialization of that started. It's the expanded bed adsorption technology. We actually developed columns, 6, 7 different sizes up to 1 meter diameter, 2 different versions of the distribution system, 7 or 8 different resins, and really worked very hard. I had a guy working for me at that time who certainly did about 100 industrial trials over the year, and most of them were actually successful technically. I was later head of marketing and one of the 2 guys who finally terminated that technology and did not continue the development. The reason was, as I phrased it back then, and God saw our turnover with this technology, and she turned around and cried bitterly. It was really not really worthwhile from a commercial perspective, and the reasons were exactly as you described. No one wanted to take the responsibility to actually put this technology onto the manufacturing floor, apart from 2 or 3 companies worldwide. So why did that happen? I mean, despite of the fact that we worked together very closely with 2 or 3 large industrial companies who helped us with the hardware development, I think from the very beginning we did not have collaboration looking at the whole concept, and we came basically out to the market as a surprise. Now this is possible. You can delete the centrifuges and you can delete all the filters and just go straight to purification. Sounded like a perfect concept, but the implementation kind of sucked. So what have we learned from this? We now run every single major project in development from the very early specification moments with someone in the industry who has the real life problems. Okay, and that has improved things a lot. However, I can still say, and I talked about that a little bit yesterday also when I spoke about continuous processing, it takes 5 to 10 years when we launch a new method, a new resin or whatever it could be, before we see significant blips on our sales curves. So one might actually ask, how do you expect that technology is going to get developed if we always have to wait 10 years? It's very different from drugs, where once the drug is launched, it takes off relatively quickly, and often gives billions of dollars of revenue after a few years in quite many cases. We live off thousands of products to make 1 billion of revenue, kind of, right? It's a very, very different game. And I've heard that question a few times. The risk averseness in the industry is so high that you really have to ask yourself 3 times before you develop a new technology in the first place. And then on top of that, mentioning single-use technology, for example, this market is very small compared to mass markets. For commodity products, there's still 5 suppliers, and every sourcing guy tells us we need dual sourcing. So basically, the market share that we could ever get is cut into pieces just by that phenomenon. Summarizing that, I would say it's really essential that technology development happens at an as early stage as possible, together with the users and with real life problems and challenges also, right? Those challenges that management will put up in front of this new technology, they should be there from the first moment. And then I think we can avoid some of this. I mean, there are success stories around resins, around equipment also, where this worked very well.
Co-development is actually the way to go, I think.
As
as I think back, I try to diagnose, why the risk aversion is there, right? And in many ways, it's understandable. And one of them has to do with the fact that people's long-term memories work a lot better with really bad stuff, right?
Indeed.
When it worked, it's great and you celebrate and things are good. But when it didn't work, boy, do they remember, many years later. So I remember every example of, forgive my French, screw-ups, right? We made a decision and it turned out to be a really bad decision. Those are learning moments. But people remember those things and they're etched in your memory, and you have a few of those, and pretty soon, you're going to be as risk averse as the next guy, especially when you're the final accountable party. So I remember wave bags, they came out, and the first time we implemented them, they looked great, and then you had the leaching problems, and the cells didn't grow quite right, and we had to trace it all the way back to the film supplier and what was going on there, and other plastic leachables, and other technologies that got implemented that didn't quite work the same way. And so I think that probably has something to do with once bitten, twice shy.
And this is a perfect example, the wave bags. I mean, the problems got solved, not the least with the help of Amgen, for example, and a few other companies. But we acquired this technology, I think, in 2004. And one of the first things that happened was that our engineers found out that the equipment, you could actually hurt yourself with it. So we had to spend 2 years re-engineering the equipment. This was invented by a very creative guy, but he did not think industrial with the technology. And there was no, how should I say, level of documentation about the plastic materials, etc., that you would need to actually detect things like this. Leachables and extractables was not really an established concept. In a due diligence, you can only find out so much, believe me. People are trying to sell their company, and they're trying to hide things. So, yeah, I mean, that's at the same time a really good example how when you then later on put effort into a technology, problems can be solved and jointly solved also. So that's really important. We also heard from Amgen about the Singapore facility and the bioreactors there, which are ours, 2,000 liters, 6 of them, or 6 workstations as far as I'm aware. And they are operated there under much tougher conditions than what they were originally developed for, which is rather dilute cell cultures, mammalian cell cultures. That was the original concept for single-use bioreactors. And when they come with these high biomasses now and long operation times, then they come under stress. So we did some co-development and fixed those problems, right? So I think it's really important that, to your point, if you want to use technology as early as possible, both sides have to be prepared to put some time into this. And with continuous processing, which I talked about yesterday, it's similar again. There are niches where continuous processing certainly has great advantages. And there are companies who are on the side of the believers, a few at least, but it meets a lot of resistance. So, if we ever want to benefit from this, there has to be a dialogue about this. We have consciously not taken the step to put systems for continuous processing onto the shelf and wait until people are coming and buy them and run big marketing campaigns about that. There's no way we could get permission, financially speaking, for doing this if the hesitance is so big, So we are basically out there having this technology tested now, and we'll see where it goes. And we don't have unrealistic expectations about that anymore, I don't think.
I recall talking about continuous processing close to 20 years ago.
Sure, it's old.
Yeah, I mean, the concept is not new, and it's really attractive, And yet here we are 20 years later.
And debating it.
And still we're debating it, right? So
Another
you need some early adopters, people that are willing to take that risk and go into the use of these technologies and essentially be the guinea pigs for figuring out the problems, the real world problems that happen, and solve those real world problems. And once others see that you are successfully using them, then the barriers come down. But the first ones to move in there, those are the difficult ones.
There's another problem with this. We have these meetings, and certain topics turn into the hot topics. So we run conferences about single use. Do we run conferences about stainless steel anymore? I don't think we do. We run conferences about actually about continuous processing. Do we run conferences about batch processing? I don't think so. Basically, certain topics become a hype for a long time, and communication, the way our world works is, someone says something which sounds right, and then it gets repeated and repeated and repeated, and after a while it becomes a self-running story, like the downstream bottleneck, for example, which, still a downstream process runs in 2 days and not in 14 days in a 3-shift operation. Where is the bottleneck? Well, the bottleneck is really one of mismatch between the installations, upstream and downstream, right? So once that problem got fixed, like in the Amgen Singapore facility, there's no bottleneck anymore, okay? And so I think we need that dialogue also. I really believe that upstream and downstream folks should sit in the same offices, in the same buildings, and not on different sites or in different buildings. I don't know how that is.
But you wouldn't have anybody to blame for when stuff happens.
Yeah, exactly. Well,
You always have to blame upstream.
ahead.
So I'd like to cut in here for a second, because as an interactive panel, terrific up there, but you guys are also part of this experience. What's that? Yeah. So you're part of the experience, and we do want to hear from you. So obviously, many of these things sound familiar. We're talking about technologies and resistance to it. Are there any perspectives, or are there any experiences in the audience here that you would like to share with us? Because we certainly like to kind of include it in the conversation. Yes.
Thanks. Dennis Boyle from Promega. A lot of companies look at the ROI for a new technology, right? So when they invest in a new technology, they're looking out. They're going to put people on this. They're going to put a lot of resources on this to make it work in their programs. And so the opportunity, if I'm right, and innovation occurs incrementally, not transformationally, then maybe the opportunity is lost. So it's kind of out of phase with the adoption of a new technology that comes along. The other piece is in clinical programs, right? So if you commit a particular piece of technology to a clinical program, as you advance that, you progress, then you have less and less opportunity to be able to take advantage of newer opportunities as well, too.
So, of course, I greatly respect Gunter's experience, but I would argue that the uptake of repetition of a message is marketing, in a way, and we've been successful in forwarding the idea. I think that my name is Lynn Frick, and I've been working on continuous processing for quite some time. I understand all the objections, and we've been very close to many customers who are thinking about this, but I do think there will be a move to a clinical batch in late 2017, 2018. And I think, yes, until you get there, there's always going to be a point at which a new technology becomes greater than just one unit operation. And it does take time, but I think this is closer now than it's taken a long time. It's already been 10 years, and so there's a point at which it finally gets going. So I look forward to other people looking into this, and I think, no, it's not the answer for everything, but one thing we've learned, I'm now with Paul Corporation, is that once we built that lab in our facility to implement only continuous downstream in one portion, the things you learn about your own products and where the gaps are and how suppliers can help the process are immense. So, you find out that, hey, a virus filter is in the wrong holder. It doesn't make it easy to manifold it. The connectors are wrong. And so by suppliers taking on the burden of doing some of that development in their own lab, in a real running process development lab, I think that goes a long way to helping our customers in offering products that you can use in PD, for instance, hooking through a PD lab still into a central data system, hooking to Delta V, hooking to Siemens, figuring out the forward, backward control, the alarm, the PAT, all of these things. If you can start to do that as a vendor and work, as Gunter said, with co-developers, then you are reducing risk for everybody. And I think that's what it takes.
This has been an excellent forum for exchange of ideas and so on. And the one comment I would like to make is, are we really closer to applying these newer technologies as compared with 20 years ago? Take, for example, your comment on continuous bioprocessing that we've been talking about for so long. It's an excellent idea, yes. I mean, it's the technology is applicable, yes. What are the hurdles we face in terms of validation issues? And I've heard so many talks on continuous bioprocessing, which have been excellent, but I haven't really heard any specific discussion of the issues in terms of what type of validation would be required from a manufacturing standpoint. Makes excellent sense. But what would I, as the user, have to do to ensure that the system is validated? Today, our emphasis is on CQA, identify CPPs, et cetera, et cetera. So how do we address those issues?
Yeah, I can make a comment on that. Is your microphone on again? Yeah. First of all, those who believe in the continuous processing, like Lynn also mentioned, there are some who are moving forward with this also on the downstream side. They say that, actually the FDA confirms that, there are no fundamental issues with doing the validation of a continuous process. In fact, FDA speakers have confirmed that and have described, how a batch can be defined. It can be by time or by volume. It's basically the user's decision, as I understand that. If I may be a little bit cynical for a moment, I would say those high-level FDA speakers who are saying this have forgotten to tell their inspectors, right? So when you actually meet an inspector, there may be lots of questions about this, and they are not very well informed about these issues. So there's a bit of a hiccup there, but I don't see fundamental reasons to stay away from continuous processing for any regulatory or validation reasons. Also in the EMA, there is a clear definition of what you have to do. It doesn't go down to the details as usual. The FDA or the EMA don't tell you what you can do and what you can't do. They just say, show me data. And that is where I think we still have a technology gap. We still don't have product sensors that tell us permanently what happens in the bioreactor or in the downstream process. How does the product right now look like? Fingerprinting, inline, online type of analysis. I think that the real breakthrough on processing technology, be it continuous or other, will only come when we have that. Because only then will we be able to say at any time, this is what right now happens in my process, so therefore I can move on and I can actually even release that batch pretty much now, and I don't work in the dark anymore.
Let me add a little bit of a different addendum, perhaps. Quite a few of our clients are in the cell and gene therapy business, right? So while cell and gene therapy have been around for a while, it's only recently that they're really starting to make good progress. And they're by necessity, because these products are showing so much promise and are being advanced very quickly. By necessity, they're advancing technologies to help that industry. And there are new problems that nobody even knew existed, right? So for manufacturing of autologous cell therapy products, for example, there are issues that just have to be solved. They're solvable, but they have to be solved. And what we're finding is necessity is the father of solving problems, right? These companies need it. They are demanding their suppliers to come up with solutions for these problems, and they're plowing forward. And there are risks that are being taken and accepted as a given, because nobody has done it before. Nobody's figured out how to validate these processes. Nobody's figured out how to reliably manufacture these products on a regular basis, et cetera, et cetera. And the problems are being encountered and they're being solved, and it's a given. With the more traditional recombinant proteins, there are ways of doing it, so it really requires taking risks for which there are alternatives, and so there is more aversion to taking risks. So it's you know.
Our industry is all about risk benefit. So when we talk about cell therapy, the risk is intuitive. On the other hand, for something like recombinant products and the plasma-derived products and so on, we have quote unquote relatively safe approaches in place. And to come back to your comment on continuous bioprocessing, I think we need to get down to more specifics. And I will give you one example. For those of you who know me, my expertise is in the area of virus safety assurance. And with continuous bioprocessing and documenting virus clearance by these during the continuous bioprocessing operations, I have yet to see a continuous bioprocess where the virus clearance capability of the particular step has been documented and provided to the regulators. If you have a general discussion with the regulator, they would say, in principle, that works, it's excellent. So, like you said, once again, it comes down to a specific manufacturer, buying into the continuous bioprocessing or whatever the new innovative technology is, and then moving forward, keeping once again in mind this whole risk benefit analysis concept. So that's what I'm saying.
Can I add something to the question? Because I fully agree with you, and I, from an operations perspective as well as from kind of the marketing application perspective, you're absolutely right. We have to go through and make sure that we can have that solid viral validation package. Otherwise, you're completely dead in the water. It's challenging. It's going to have to be different, essentially, from what we've done, because we always have one pool, we go to another pool, you test your starting pool, you test your ending pool, you're good. But now we don't have a starting pool or an ending pool. So we have to be a little bit more creative. We have to do things in line. Unfortunately, we don't have all the analytical techniques of inline techniques, but I know from Amgen's perspective, we've come up with ways of doing things, quote, on the fly, and getting those representative samples to show that, yes, your starting and ending material did show sufficient clearance. It's just not going to be through your traditional means.
How do you demonstrate scale down? Until you take it offline.
Sure.
Because I don't want to take it on.
And
Hey,
it's challenging.
so I'm Matthew Stork, and so I work in process development with Pfizer. And so I actually just had a comment kind of related to, resistance to change. And I totally agree with the previous comment that in many cases it really is just about marketing. So in process development, I think the barrier to introducing a new process development technique, in my own personal experience, I do a lot of high throughput processing, but the barrier is fairly low. And if you screw up, well, you've just lost a week of your own time. But even then, there's still some resistance to going and adopting these sorts of techniques. So, I have really found that the marketing aspect in my own personal experience has been quite important. And along with that, I feel like resistance to change can often be due, I guess, to siloing of innovation, where you might have specific technology initiatives. And within those initiatives, everybody is very willing to go and work on these innovative techniques and stuff like that. But when it comes to actually doing projects, well, the bar is much, much higher. And, there's, not much willingness to actually adopt new techniques. So I know on Monday, we had heard at the continuous processing symposium some techniques, I guess, for sort of marketing new ideas. But I was wondering if any of you had any perspectives on that.
Maybe I make a comment on the continuous processing. There we have the continuous upstream process. And that process is, let's say, several months long. And when you validate that several months long. Several buildings, several suites, several scale-ups. You have to do all that to show that the product from the beginning, middle, and end are the same. So there's no way around it. So I think people who propose that we do continuous processing seem to forget that. When you want to make a change, it's very difficult to validate. But for us, we have to do it because our product is unstable otherwise. So I always feel continuous processing, we should do it when we have to. I love batch processing, by the way, now. I've worked on it for a while. But just one more comment on, this conversation has gone to new technology. Again, I say if you can show that this reduces reject rates, it's always going to fly. And return on investment, if you can say this new project or new approach will fit your process into two buildings instead of 3 buildings. Very simple. There's no need to calculate anything. The management will immediately buy into it.
Thank you.
You know, one of our challenges is that the people that understand the technology the best are the scientists and the engineers. I certainly don't want to be generalizing about everyone because I'm an engineer by training, but scientists and engineers don't tend to be very good communicators. So maybe that has something to do with our marketing problem on new technologies. Although Catherine's an excellent communicator.
Thank you. I was asked to present kind of new technologies at one point to some of our executive vice presidents, and at a very basic level, because some of them have the scientific background and understand the details, and some of them just don't, and they don't need to. I mean, they're at a very different level and do a very different job function than I do. But at a very basic level, I took a column, small scale, and I said, well, we're trying to convert to this flow-through device that's this big. Just 2 different sizes, and they went, wow, that's fantastic. Is it really that difference in size? I said, yes, and you throw this away, and you'd have to keep this and validate it and reuse it and all of that. They said, oh, go do it. Okay. So what I find is getting buy-in for that, keep it somewhat simple. You do have to tie it back to the financials all the time. But from that perspective, it was quick, it was visual, and everybody got the point very quickly.
Although I do have to say that you better hope nothing goes wrong because they will remember.
Absolutely.
And I've been there. I've been there. Honestly, I remember being in meetings where, you know, I described here's what needs to happen, and no. If we do it this way, you'll save $10 million. Yes, but there's a risk of failure that you can't overcome. Yeah, that's okay. We're willing to take the risk, and we went that route, and it did fail, and none of them remembered saying we are willing to accept the risk. Right. So what part of the risk taking did we not understand? Was it we're willing to take the risk as long as nothing goes wrong? That's not taking risk. You know, I do give them credit. One of them actually remembered and said, yep, we said that.